The overall goal of this workflow is to identify certain targets for ultrastructural imaging within a tissue using light microscopy followed by 3D imaging in an SEM at very high resolution.
The imaging workflow introduced here can help answer key questions about cellular or tissue ultrastructure in a number of fields such as cell biology, developmental biology, neuroscience, or even pathology. The main advantage of the technique, which is actually based on array tomography, is that slicing up tissue into arrays of serial sections makes it easy to identify target structures, even when initially they are buried inside the original sample volume.
Array tomography was originally introduced in a neuroscience context, but it can also be applied to a wide range of other systems, including bacteria, plants, animals, and even patient samples in a pathology setting.
Individuals new to this method will struggle, because collecting many serial sections is very difficult. Working without additional support may cause loss of sections or disarrangement of the ribbons.
Using a tiny brush formed from a few hairs fixed to a toothpick, carefully coat the leading and trailing sides of a pre-trimmed block with an adhesive mixture. Perform this step quickly because the solvent of this mixture evaporates within seconds. While the samples dry, cut pieces of silicone wafers to a size that fits into the knife boat and mark them with a diamond scriber. Then, clean the silicone wafer manually with isopropanol and lint-free tissue. Fix the substrate to one end of the carrier plate using a removable adhesive. Once set, plasma activate the substrate using glow discharge with air to obtain a high